Translational Medicine Day Committee. I will be your host for this session, in which our speakers today are Dr. Hannah Carter and Dr. Debashis Sahoo. So just as a disclaimer, we will be recording this session, so feel free to turn your videos off and mute your mics. So now I'm going to provide a brief overview of the purpose of these speaker sessions. So we want to educate the public. So we want to educate the public and increase awareness about the research within this rapidly growing field of translational medicine, all while highlighting the variety of disciplines within the field itself. So with that, I will provide a brief introduction of our speaker number one, Dr. Hannah Carter. So Dr. Carter is an associate professor in the Department of Medicine, Division of Medical Genetics. Her research uses computation to analyze patterns of genetic variation in host and tumor genomes to advance precision medicine. Specifically, her lab is studying tumor genomes to learn about genetic factors contributing to cancer risk, progression, and response to therapy in order to advance personalized risk prediction and uncover novel therapeutic targets. With that, I will hand it off to Dr. Carter.
Thank you for the introduction. I'm just going to share my slides. Okay, so it's my pleasure to have the chance to share some of my research with you today on Translational Medicine Day. So our lab is interested in precision medicine in the context of cancer specifically. Just to give you kind of a high-level overview of what that means for us, we're interested in interpreting the inherited or germline genome in the context of individual risk of developing disease, with an eye to identifying groups at higher risk that could benefit from more effective early detection to prevent development of cancer. And sometimes these germline variants can provide clues as to preventative strategies we can use to help prevent people from developing cancer. Then on the flip side, we have the somatic genome. So this is the genome of a tumor once it's already established and been diagnosed. So if you extract a tumor and sequence it, you can identify all of these changes relative to the inherited genome. These are what we call the somatic genome of the tumor. And it tells you a bit about the mechanisms that have driven the development of the tumor and can provide sort of clues about therapeutic sensitivity. So we'd really like to take characteristics of the tumor genome and use it to stratify patients according to the optimal therapy available for them so that we can improve patient outcomes. Something that my lab particularly has become really interested in is studying how the germline interacts with the somatic tumor genome. This can provide us further clues about the mechanisms that predispose to cancer and point to specific types of cancer driven by particular somatic alterations, which then can tell you maybe even earlier if someone develops cancer, what kind of cancer are they likely to develop. So just to give you a little bit of background, we extract the tumor, we sequence it, we identify the differences relative to the germline genome. These are the so-called somatic mutations that characterize the tumor. We've done this for many, many tumors now. We have these great databases like the Cancer Genome Atlas that has 10,000 different tumors sequenced according to the somatic changes. And we found that if you study the profiles of somatic changes, you can identify information about the history of the tumor, such as the types of mutagenic exposures it's been subjected to, as these have sort of specific fingerprints that are present in the DNA sequence. And it also tells you something about the evolutionary history of the tumor. So tumors develop typically over decades, and the sort of the model that we usually think of is one where you have the sequential accumulation of driver mutations that lead to these sequential expansions of cell populations that are increasingly developing neoplastic characteristics that drive to the malignant disease. And this process is thought to reflect a predominantly positive selection process that's acting to fix driver mutations that provide a fitness advantage for the cell population. So this particular talk is about some studies that we've done of the inherited genome in the context of the immune system. And so why focus on the immune system? Well, first, this picture on the right depicts what we call the so-called hallmarks of cancer. These are characteristics that are developed by all neoplasms. So all cancers develop these characteristics at some point, and that suggests that these are in response to some kind of selective pressure acting on the tumor. Notably amongst these is avoiding immune destruction. And in about 2013, there was a breakthrough in this field of checkpoint inhibition. But basically, conceptually, the tumor adopts a mechanism to evade the immune system. And if you can impair that mechanism, you can get robust killing of tumor cells by the immune system. So it's pretty clear immunity is placing selective pressure on the tumor. And that's a place where we can look and see if genetic variation influences that, that relationship between the pressure from the immune system and the evolution of mechanisms of immune evasion by the tumor. So just to give you a primer on the anti-tumor immunity, we're focused on this particular mechanism of antigen presentation here. So this, imagine, is a tumor cell, and proteins are translated inside the tumor cell. If they have mutations, they may be driving the neoplastic characteristics. All proteins, mutant or not, are going to be degraded into peptides and bound to this molecule called the major histocompatibility complex, MHC class 1, which is expressed on all nucleated cells. And then in complex, the peptide in that protein complex will make it to the cell surface, and the peptide will be displayed to CD8 T cells as part of immune surveillance. The reason that this doesn't trigger killing of our cells normally is because peptides that are self are expressed in the thymus, and any CD8 T cells that recognize those peptides in the thymus will get pruned from the repertoire. So you shouldn't get any reactivity. But the mutants are not expressed in the thymus. So mutant proteins in the tumors have the potential to activate TCR, CD8 T cells in the T cell repertoire, which can result in killing of the tumor cell. So this major histocompatibility complex then is a key player in this process and kind of a bottleneck. If you don't have this, CD8 T cells cannot detect foreign antigens in tumor cells or attack them. So MHC class 1 is expressed on all nucleated cells and predominantly expresses presents peptides from proteins that are intracellular to CD8 T cells. And the peptides tend to be on the length of 8 to 11 amino acids long. This is encoded by the HLA locus, the human leukocyte antigen complex, on chromosome 6. And so we're going to ignore class 2 today and just look at class 1. These 3 genes are encoded by over 14,000 different alleles in human populations. So there's this huge diversity. And so any individual can have very different versions of each of these genes. So sequence, as you are probably familiar, is a determinant of structure in the protein world. And so different sequences of these alleles can mean slightly different structures, specifically of the peptide binding groove of MHC. And differences in that peptide binding groove structure are going to change the specificity for peptide antigens that it can present to T cells. What that means is that based on your individual collection of HLA alleles, you may have MHC with a particular specificity for the domain of possible peptides that can then be presented on the cell surface for surveillance by T cells. So the work I'm going to describe now was by a former graduate student, and now a PhD from my lab, Rachel Marty, and a former postdoc who's now faculty at the Fox Chase Cancer Center, who evaluated the hypothesis that if we look at what the inherited MHC genotype presents, and we overlap it with cancer-causing mutations, then you can imagine the scenario: if a cancer-causing mutation generates a peptide that's effectively exposed by the patient's MHC alleles, then it would be subject to immune surveillance, and potentially the tumor cells can get eliminated by T cells. Whereas if that cancer mutation doesn't generate any peptides that effectively bind any of these, then it sort of can drive proliferation of a cell population without any alert of the immune system via the antigen presentation pathway. And so you would expect these to occur with much more higher probability in tumors. So we set out to test this hypothesis, and the first thing we had to do was develop a computational pipeline for predicting whether or not a mutation was going to generate a peptide that could be displayed on the cell surface given a candidate or a set of MHC alleles. So you have your alleles, you have your mutation, and you're going to look at all possible peptides overlapping this mutation. relative to all of these alleles. And we use a tool called NetMHC-Pan that gives you a binding affinity of peptide to specific allele. So for each allele, you can identify the peptide of length 8 through 11 amino acids that would best bind to that. And then, there's a sort of a threshold of where binding is strong enough to actually have a stable complex on the cell surface. And most alleles may not display any peptides, but you may find different peptides for different alleles. So then we can take this rank for the best peptide for each of the alleles. And here we want to aggregate the effects of all alleles because all alleles have a chance to present the peptide on the cell surface. And we ended up using a harmonic mean because a low rank indicates presentation, whereas a larger number would indicate that the peptide probably doesn't have strong enough affinity to make it to the cell surface. So we want to prioritize lower scores. That's what the harmonic mean will do in this case. Low score means higher probability of display on the cell surface. And then we could evaluate whether or not this actually is representative of the likelihood that something will make it to the cell surface by comparing to these mass spec data sets where they elute the MHC complex and the peptides and sequence the peptides. And then we can compare to the random presentation of random peptides versus what's actually eluted from the MHC. And you can see that you do a reasonable job of predicting what's bound and displayed on the cell surface just based on the combined affinity across the multiple alleles alone. Definitely room for improvement, but you can do some prediction of what's displayed physically on the cell surface. So now that we have this tool in hand, we want to test our hypothesis in a tumor population. We went to the Cancer Genome Atlas, which has about 10,000 tumor genomes in it. And based on these, we can use bioinformatic tools to back in their HLA type. So we need to take the sequencing data and predict which of the 6 alleles each person, which alleles each person carries at the 6 loci. And then we wanted to evaluate whether this has an effect on the affinity profile of the mutations in the tumor. However, we know that all tumors develop the ability to evade immune detection at some point. And so we expect that mutations that occurred late are less likely to be constrained by HLA genotype than mutations that occurred early during tumorigenesis. So we wanted to emphasize mutations that were likely to be early drivers of disease, as those would be the most interesting to study in the context of immune selection. So we looked at recurrent somatic mutations in 100 known oncogenes and 100 known tumor suppressor genes, resulting in about 1,018 mutations. And now we can calculate for nearly 10,000 cancer patients, given their HLA genotypes, what is their potential to display a peptide covering one of these mutations on the cell surface for immune surveillance. And so that's what this is telling you. In this heat map, each row is a patient and each column is a mutation. And here you can see a darker color indicates that their particular HLA genotype could effectively present one or more peptides that overlap that mutation. Whereas a lighter color would indicate that they were not able to present or display a peptide of that mutation on their cell surface. And you can immediately see there's a couple of mutations that look like they would always be displayed, and a couple of mutations that look like they would never be displayed, but then considerable inter-individual variability for other mutations. Now, we didn't really find any clustering of mutations on the basis of the type of gene or occurrence they were, or any clustering on the basis of whether the tumor was immune infiltrated or not, but we did see some ancestry based clustering, which isn't surprising because the allele frequencies of specific HLA alleles differ by different ancestral groups. So now what we want to ask is, if you have a mutation that is well presented, is it less likely to actually be observed in the tumor? So we're going to mask this, dividing it into 2 components, one when a mutation was observed and one when a mutation wasn't observed. And that's what this looks like. And just to clarify, the average tumor carries somewhere between 2 to 8 driver mutations. And in many tumors, we cannot identify what the driver mutation is. So we do expect this to be really sparse. But if we collect the presentation scores where a mutation occurred versus where it didn't occur and compare the distributions, we can see a shift actually right around the binding threshold of 2 for mutations that were observed to have a bias towards higher scores, indicating that they're less well presented on the cell surface. So this seemed consistent with our hypothesis. We can be a little bit more quantitative about it though. So we used a mathematical model. And so here's the sort of the sparse binary matrix of whether or not a mutation was occurred. And now we're relating it to the potential to display on the cell surface. And we're also controlling for the mutation frequency in the individual tumor genome. And the question is, then is this coefficient going to be significantly different from 0? And if we convert the value to an odds ratio, what we see is that for mutations that are sort of recurrent driver mutations, they are more likely to occur when they're poorly presented. So that would be consistent with our hypothesis. But we don't see the same for random one-off passenger mutations, nor do we see it for germline variants, which is consistent with this being specific to these oncogenic mutations. So then we can break down and look across different tumor types to try to see if this is sort of generalizing across multiple tumor types, or if particular tumor types maybe drive the phenomenon. And you can see that actually the majority of tumor types we assayed, we found a significant bias for mutations to prefer to occur when they're less well presented. The strongest effects were in thyroid cancer and low-grade glioma, but those are driven by a very simple mutation profile, usually with thyroid cancers typically always having BRAF V600E, and low-grade gliomas usually having IDH132 mutations. And those two types of mutation tend to be generally poorly presented by MHC, as I'll show you in a moment, and that might be driving sort of these inflated effect sizes for those tumor types. But it does seem to generalize in a lot of cancer types, and we also see some exceptions. It's not entirely clear if this is due to sample sizes or if there's actually something really going on there. So it's something that we're keeping an eye on. So here we've now ordered our cancer causing mutations according to the level of recurrence. So you see the least recurrent mutations here and the most recurrent mutations here. And then each row is once again a patient, and you're looking at their average ability to present mutations or peptides from mutations falling into this sort of bin of recurrence. And you can see this shading from darker on the left to lighter on the right, which suggests that as it becomes more difficult to present mutations or display peptides for mutations on the cell surface, those driver mutations tend to be more frequent in general across the tumor population. So it's a little bit disappointing in that it suggests that if you could find mutations or neoantigens that would be shared along across a large number of tumors, it would be hard to develop an immune vaccine against those because they're already sort of predisposed to not be very immunogenic in the tumor setting. And we do see some exceptions, but and those look like they might be interesting, but in general, this might just suggest that there's some other reason why it's not well presented besides affinity, because it's hard to imagine that you could have a highly recurrent mutation that's also highly immunogenic. Okay, so let's talk about translational aspects of this then. In the clinic, we're really interested in using immune checkpoint blockade to kill tumors. And so the concept here, you have your MHC complex with your antigen that's activating the T cell. But the T cell has these sort of co-stimulatory receptors that it expresses, and some of them are inhibitory, like PD-1. So if the tumor cell expresses the ligand for this, PD-L1, it's actually going to shut down the activity of the T cell. So what immune checkpoint blockade does is you use an antibody to either block the PD-1 or the PD-L1 side of this interaction. Now you can't have this negative signal given to the T cell. The T cell is going to now eliminate the tumor cell on the basis of the signal. So it's an exciting possibility to rejuvenate the immune response against the tumor and have your own immune system eliminate it. However, success has been limited. For the best cancer types, we see maybe 40% of patients respond, and many end up developing resistance even to the immune checkpoint blockade. And we're trying to understand what predicts who's going to respond and try to understand why people don't respond. Well, the earliest measure of response, here we're talking about durable clinical benefit versus no benefit, is the non-synonymous burden of mutations in the tumor. So having a higher burden of mutations and potentially neoantigens seems to be predictive of better outcomes in cohorts treated with immune checkpoint blockade. And this has sort of been general over many different tumor types that have been studied. You can see that mismatch repair deficient colorectal and non-colorectal cancers and skin cancer, where you tend to have a lot of UV driven mutation and melanoma as well. These are places where you have a very high mutation burden, and they tend to generally have the higher response rates to immunotherapy. So we collaborated with clinicians at the Morse Cancer Center to apply our paradigm looking at how well mutations are presented by an individual's MHC with whether or not they were likely to respond to immune checkpoint blockade. And so here are 77 tumors that were treated with immune checkpoint monotherapy, and it's from diverse tumor types. And you can see that if you just threshold on tumor mutation burden, you can sort of stratify the patients according to outcome. What we found then, though, is if you overlay onto that whether they're able to present one or more mutations in their tumor genome with a very high binding affinity, then we can further stratify this high tumor mutation burden group into those that are going to respond and those are not going to respond. So tumor mutation burden alone is not enough to guarantee a response. You need to have a high tumor mutation burden that includes an effectively presented neoantigen, essentially. In our more recent work, we've been trying to expand this beyond the MHC. So Megan Apagadala, who's an MD PhD student in my lab, recently performed a screen. It's essentially a GWAS, but instead of looking at cancer versus non-cancer to identify germline determinants, what she did was look for heritable characteristics of the tumor immune microenvironment and identify the germline variants that were associated with having different characteristics of the tumor immune microenvironment. And she identified 482 variants associated with various characteristics and was able to back about 69 of those into a set of ICB cohorts. And so these were all sequenced with exome sequencing. They didn't have SNP chips. And so we had to impute SNPs at these key germline variants from the exome sequencing data. And then we could study the relationship between those germline variants and whether or not people respond to immune checkpoint blockade. And this is just showing that even just germline variation that influences the tumor microenvironment is predictive to some extent of whether or not someone responds to immune checkpoint blockade. Now, we don't expect a huge level of predictive activity of the germline. We would expect the somatic profile of the tumor to be more predictive. And so we're integrating the somatic and germline factors together to boost performance here. But what's really cool is that these germline variants that are predictive of performance are also, many of them are EQTLs in particular immune cell types and whatnot. And so they're revealing sort of new targets that we might be able to manipulate in order to maybe prime the tumor immune microenvironment and try to improve response to checkpoint blockade. So that's sort of a new translational angle that we're going after based on this research program. So just to summarize briefly, we talked about the MHC allele combinations that each person carries and that these are sort of the determinant of how well mutations can be exposed to an individual's immune system. That mutations are more likely to be observed in a tumor if they're poorly presented by the individual's MHC and invisible to their immune system. And across large tumor populations, what this results in is that the most frequent mutations are the ones that are on average less visible to the immune system across multiple individuals. That being said, effective presentation is still a determinant of response to immunotherapy. And in our follow-up work, we're finding other characteristics of the tumor immune microenvironment that contribute to response to immunotherapy. So we have a little bit of a conundrum in that we have this tool that has a lot of potential to effectively eliminate tumors, and yet tumors are actively evolving to try to evade the immune system that we're turning against them. This might explain in part why we only see some folks responding to immunotherapy. And certainly loss of MHC is a mechanism of immune evasion that's not that uncommon, as we're finding frequently individual HLA alleles are deleted by copy number variation. Okay, so this work wouldn't be possible without an amazing team and a lot of fantastic collaborators, both at UCSD and elsewhere. And we're very grateful to our various funding sources that support the work that we're doing. Just a shout out to Rachel Marty, who did the first part that I talked about, and Andrea and Megan, who worked on the translational studies that I described. So I'll stop here and thank you for listening, and I'm happy to take any questions.
Thank you so much, Dr. Carter, for your presentation. So if you have any questions, feel free to unmute your mic or type it in the chat. So there's a question in the chat. Has your study seen any difference in the connection between mutation and tumor growth across gender lines?
Yeah, that's a fantastic question. So what we recently published a paper just this last year. Andrea was the lead author. And what she looked at was whether the relationship between the genotype and the probability of observing a mutation was equally strong in males and females, as well as in young and older individuals. And what we found was that the relationship was stronger in younger and females. And basically what it suggests is that the pressure of the immune system on the developing tumor is stronger when you're younger and have a robust immune system, but also in women, which is not maybe that surprising, as there's a lot of immunological differences associated with, pregnancy and things like that, and have generally been associated with a stronger immune response in women.
I think we have another question. So earlier you mentioned a technique called screen. What is it and how does it compare to a GWAS?
Oh, I was probably referring to a genome-wide association screen. So basically, we're looking for association between a large number of SNPs and in a large number of individuals across a particular trait. In this case, we looked at, I think we started with something like 700 immune traits we thought looked interesting, but then only about 300 of them ended up having any evidence of heritability across the 10,000 tumors. And so we only did the genome-wide association study for SNP associations with the traits that were heritable across those 10,000. And then we had to penalize for the number of traits that we examined, but we still found something like 482 associations that were significant.
Are there any more questions?
I have a quick question. So I thought it was really interesting how you looked at both like somatic and germline mutations. So when you're looking at like somatic versus germline mutations, do you find like the results and trends agree with each other? Like an individual who has like a very aggressive cancer will have both germline mutations and somatic mutations, or like do you ever get a point where they're like conflicting?
So that's a good question. I don't know that we've looked at it quite in that way before, but we do sometimes see that you can have a germline effect or a somatic effect. And it can be, one person maybe carries the germline and one person carries the somatic, but the tumor has sort of equivalent loss of a particular key function. So we just published a paper on TP53, and there's a 3' SNP in the UTR of TP53 that we found is inversely associated with breast cancer subtype and ovarian cancer subtype. So if you have the minor allele, you're more likely to get triple negative breast cancer than luminal breast cancer, for example. And if you examine that SNP, it's a microRNA binding site, and it's associated with reduced expression of TP53. So patients are less likely to get somatic TP53 mutations if they have that UTR SNP. And so you can kind of co-analyze them with patients that have TP53 loss relative to patients that are truly wild type at TP53. So in that case, you have like a SNP that's totally redundant. Now, I do expect that we'll see burden of SNPs associated with particular oncogenic pathways will be associated with whether or not you get activating mutations in those pathways, as well as with risk, age of diagnosis, and what type of cancer you'll develop. But it's actually kind of hard to study at this point in a systematic way because there's only really the TCGA where we have a large number of tumors that have both germline and somatic profiles available. So we're always looking for new data sets to come out, but it's pretty expensive, I think, to do that kind of effort where you sequence the tumor, you sequence the normal, you do RNA sequencing, you characterize methylation, and all of these different things. But it's valuable data where it's available.
Thank you. I just found that super interesting, like the systematic approach to it, because cancer is like a very systematic disorder.
Thank you. Yeah, we're excited to continue pursuing this, and we're interested in looking at other hallmark behaviors besides the immune system as other places where we think germline variation could modify selective pressures acting on evolving tumors.
All right. Thank you so much, Dr. Carter, for your interesting presentation.
Sure.
So now I think we can move on to speaker number 2, Dr. Debashis Sahoo. Let me briefly introduce Dr. Sahoo. So Dr. Sahoo received a Bachelor's of Technology degree in Computer Science and Engineering from the Indian Institute of Technology, Kharagpur in 2000, and the MS and PhD degrees in electrical engineering from Stanford University in 2003 and 2008, respectively. He is currently a joint assistant professor of computer science and engineering and pediatrics at UCSD. His research interests include the analysis of biological systems and formal verification of hardware designs. Dr. Sahoo pioneered identifying simple Boolean relationships between gene expression values. Dr. Sahoo's research has produced impacts in understanding several biological systems, including B-cell differentiation, colon cancer, bladder cancer, and prostate cancer. Among the successes was Dr. Sahoo's previous work, where he identified the expression level of CDX2 as a predictive biomarker for favorable response to conventional chemotherapy among stage 2 colon cancer patients. So with that, I will hand it off to Dr. Sahoo.
Okay, thank you, Rishan. Thank you for inviting me to this group. It's really exciting to see a youngster um, watching the state of the art translational research. Can I share my screen?
I made you co-host, so you should be able to. Yeah.
Okay. Yeah. All right. So I'm going to talk about. Let me put my thing here. Okay. Translational medicine using Boolean analysis. So it's basically a very simple mathematics. I'm going to describe briefly and show how it can be used to understand complex diseases and identify important biomarkers. Okay. So Boolean analysis basically a simple mathematics of two values. I call it Boolean. George Boole started this long time back, and the two values can be high or low or one or zeros. And there are the standard Boolean operations and or and not implications. These are really basic fundamental building blocks of what we use regularly, all the computers and digital circuits. I'm using this kind of simple logic to dive into this massive amount of data that we generate in the biomedical field, and try to come up with very simple patterns that can give us insights into diseases. So I'll talk about this simple logic called implication, which is X implies Y, and expand on that a little further because we're going to use that heavily in this talk. So X implies Y is a Boolean formula, right? X and Y are Boolean variables. So these variables, X and Y, they will take two values, either zeros or ones. So if you have two variables, there are four different possibilities. And if you have to evaluate X implies Y under these different conditions, you can see that the only situation where X implies Y is false is when X is 1 and Y is 0. So in an English sentence, you say if X then Y, or if X is true, then Y has to be true. So in order to falsify this statement, you have to set X to true and Y to false, or set X to 1 or Y to 0. And that's the only way to falsify the statement. If X is false, the statement is vacuously true. You can see if X is 0, X implies Y is actually true. So this is really interesting, and people have used this very cleverly in, you know, throughout politics as well. You might have heard about this phrase, if the president didn't commit a crime, we would have said so, from Robert Mueller, right? So it's not directly implicating the president, but saying in a precise way, in a way that, whatever the data they collected, this is exactly how they would have represented or summarized the data. Um, and also there are other examples, but let's dive into data analysis in this field, biomedical field. So if you look at data, they come in different varieties, right? And here we are looking at gene expression data. The gene expression data is basically a quantification of mRNA, right? And the way you measure it is using microarray or RNA-seq. And at the end, these values are summarized into a range of continuous values. So they are not Boolean, but if you have to perform Boolean analysis on gene expression data, you have to somehow divide them into two values. So one way to summarize, one way to divide these gene expression data into two values is to sort the values for a particular gene. So you look at one of the gene, CDH, for example, in this case, sort all the values from low to high and fit a step function. And so you find the threshold based on the step function and come up with the high and the low values. And around the threshold, you demarcate a region that you consider like noise, like this is intermediate values, but they are not interesting from the analysis perspective. After that, you look at a pair of genes, and this is very easy to look at because it's two-dimensional, it's easy to visualize, but you could do this on a much higher dimension computationally. If you look at a pair of genes and overlay the threshold on these genes, what you have is these 4 quadrants. And if any one of them quadrant is empty, it follows a logical pattern that you can summarize using a Boolean formula. For example, on the top left side here, you can say if PTPRC is low, then CD19 is low, which is also equivalent to saying if CD19 is high, PTPRC is high, which means this top left quadrant is sparse. So in order to falsify the statement, right, we have to set PTPRC to low and CD19 to high. And that is precisely what is the false this sparse quadrant is. So it's rarely, so it's extremely rare to see points in the sparse quadrant. And based on the sparse quadrant, you get all different kinds of Boolean formulas. But these are the two interesting things that everybody looks at now based on the methods that they use, which is correlation or regression strategy to establish relationship between genes. And that's the most common method. And there you use, if you use Boolean analysis, you get 2 sparse quadrant, and you call this equivalent here and opposite here. Most of the relationship, if you look at, in an unbiased way for all the gene pairs, most of them will fall in this category. Very rarely you see these. But the state of the art is that everybody is focusing on these kinds of relationship and associating other or these are integrated into all the pipelines that we see these days. Slowly, I'm trying to, um, motivate them to use other kinds of relationship and try to come up with a better approach, integrating these to understand biology. So these are, this is the mathematics behind it to identify these kind of relationship. A computer can look at these data and spit out what is the Boolean implications based on this data. Another thing you might consider is these data sets can be in different platforms or different species. So the one that we were looking at before is from human. But if you look at this data in different species, there are some relationship that might hold across all the species. For example, this one, you have a relationship between pCNA and MCM2, which is equivalent, and it seems to be similar in human, mouse, rats. These are plants, and this is fruit flies, and this is a marine organism. So across so much of different diversity, different sample types, different microarray platforms, you see that there is this relationship that seems to hold in all these data. We call these, when you come up with a formula, a Boolean formula that holds across all of these diversity, we call it invariant. Invariant means things that doesn't change, right? So we have a Boolean formula that doesn't change. That means it's kind of a universal logic and a rule that everybody follows, not just human, right? So you have mouse, rats, plants. And I'm going to give you an example of how we can use these concepts and the Boolean implications, invariants, to understand complex diseases and identify important biomarkers. One of the challenges in translation, like translating information from bench side to bedside, uh, is reproducibility. Uh, we cure mice, we solve problems in the bench side, look at important cell types and their implications. But oftentimes these informations, when we try to translate to the bedside, which is in the clinical domain, they don't hold. Very rarely they hold, and in those cases they hold, we get the benefit, right? So how can we improve that process? If you have a reproducible mechanism, for example, if you focus on invariants, the invariants help you translate this information in a much better way than other approaches. So I'll give you one example in that context. So we looked at colon cancer and try to identify these very strong, robust pattern that looks like invariants. So this is one of the invariant, and you can see if CA1 is high, KRT20 is low. It's not just true in this data set. In fact, you look at any colon cancer anywhere in the world, you will never find a colon cancer which is top left quadrant. That means it probably doesn't exist. A colon cancer which is CA1 high and KRT20 low. I can say that with a very high confidence, and it's probably not, it's not possible to have that cancer at all. And there is a reason why, and this reason is because of some intrinsic biology, and in this case, this is a very simple explanation of why this occurs. So it's because of differentiation. differentiation is a process. It's a very complex process, but it's there in tissues. There are stem cells and there are differentiated cells that perform important functions. And during this differentiation, there are key genes, marker genes that change, and they are irreversible. And they change in different order. So in this case, KRT20 turns on first and then stay on. CA1 turns on and stay on later. So if you have this model, you will never have a situation where CA1 is high and KRT20 is low. And that is precisely what we see in the cancer data. So using this kind of principle, we thought we could get some insights into colon cancer and identify important biomarkers. So let me give you an example. So we will use these Boolean formulas to look for the stem cell genes, which the genes that are very close to the stem cells. They're either expressed in the stem cells or they're not expressed in the stem cells. And so we set up these search criteria and look for which genes would characterize these kind of states. And there is all of these formulas that you can derive. And for one of these, the X, we get very selective, set of genes. So we got around 16 genes. So these genes are supposed to be genes that are expressed when the cells are differentiated. So if it's low, it is more like stem cells. If it is high, it's more differentiated. So among these, we have one of these genes that were really interesting because there was clinical grade antibody available. So you could actually look at tumors with antibody staining and characterize them with high and low. You can see CDX2 positive colon cancer and CDX2 negative colon cancer, black and white. And you can see that CDX2 negative colon cancer have worse outcome compared to CDX2 positive. There's 50% survival versus around 70% survival for 5 years overall for colon cancer. So hypothesis is that CDX2 low is high risk. Now, how can we use it in the clinic, right? We can zoom into one specific population called stage 2 colon cancer and show that we can maybe identify high-risk patient there. And the key criteria for zooming into stage 2 is that these patients are not treated with chemotherapy. So there is additional options for us to maybe give chemotherapy to these high-risk patients and see if they can benefit. So what we saw was that among the stage 2 colon cancer, if you look at CDX2 negative cancers and the cancer that the persons who are not given chemotherapy, they have 50% survival after 5 years. But if a cancer patient in this group is given chemotherapy, they have 90% survival. So this is clearly a big benefit, but you don't see that benefit in the CDX2 positive group. So this was a clinical trial that was done 25 years back, and at that time they were not able to separate these, right? They were looking at these two together, and they saw that there is no benefit for colon cancer when you give chemotherapy. So because of that, there was a clinical guidelines set, and it's still now that stage 2 colon cancer should not be given any chemotherapy, right? So right now the clinical guideline says that you shouldn't give chemotherapy to stage 2 patient, but this clearly shows that the patient who are CDX2 negative benefits from chemotherapy, which is something people have been trying this for last 25 years. They're trying to identify biomarkers to identify high risk patient from stage 2, and none of them succeeded so far. So this is the first study that shows that you could actually go from very simple mathematical formula and try them on this large scale and succeed. It's very rarely you find this, right? So people have been trying this for hundreds of times, but none of them succeeded. So we were the first one who saw this benefit. We have been trying to use this Boolean network in a variety of diseases in complex situations. I'll give you one quick highlights from IBD. IBD is a complex disease. It's inflammation of the colon tissue. It's upset stomach mainly, right? And there's two forms, UC and CD. What we can do here is we develop these Boolean networks, right? And try to predict targets and therapies and efficacy. And let me give you how far can we go with this. So think about the targets that people have tried already before. Okay, IBD, in IBD, several different have been tried, about 21 of them, right? Only 5 of them was approved, and all of these failed. And people try them because there was, a case that shows that there's evidence that shows that they actually differ from healthy to disease, right? Differential expression analysis show that all of these are differentially expressed from a healthy to a disease setting. And also when people perform complex network analysis, Bayesian network analysis, and they find the most important target in the IBD, they actually fail to identify these targets that actually pass phase 3 clinical trials. But if you do Boolean analysis, what we see is that there is a model that actually 100% predicts the targets that actually pass phase 3 clinical trials. So this suggests that you could actually, develop mathematical models that can give us some insights into how far can we go in the clinical side. So it depends on the biology, but these reproducibility and strong relationship that we see under this Boolean analysis help us tease apart these complex situations. So I'll summarize. What we discussed was Boolean analysis that discovers invariant relationship between genes. Right? Invariance means things that do not change, something that is fundamental, right, and core to biology. It's invariant analysis provides a reproducible framework that helps translate from bench to bedside. It also provides a new way to identify therapeutic targets that are more likely to pass phase 3 clinical trials. And of course, it gives us insights into the biology and come up with new hypotheses on complex diseases. So that's all, and I have a lot of people to thank. So there are many people in my lab and my collaborators, but I'll take up questions for you guys.
Thank you so much, Dr. Sahoo. So feel free to type it in the chat or unmute your mic if you have any questions.
Hello, Dr. Sahoo. I found your talk like super interesting, especially how you're looking at it from Boolean logic. And I find it like a really robust way to look at different data sets. So I was wondering, like, when you're speaking of mathematical modeling, when you're looking at like all these invariant relationships, can you say like, can you make like some sort of machine learning model where you sort of like look at a person's expression and then from like the invariant relationships, sort of from a machine learning model that you train, come up with like personalized risk factors for someone?
Yeah, that's a very interesting question. We actually did that for the IBD. So you could develop this Boolean network, Boolean implication network, and it's really big, massive. So how do you find the important stuff in there? Machine learning, right? So you have some data, you try to see which parts of the network can give us interesting insights into the disease, and you can set up a machine learning framework to zoom into the right portions there. And once you find that something is reproduced, something really strong and looks good, you can see if it works in your validation data set. And most of the time what happens is these invariants that you see, they are supposed to be fundamental, right? So those strong fundamental relationship helps you validate in unknown data sets in an easier way than other methods, right? The other thing is personalized therapy. Personalized therapy is something that, depending on how it translates to the clinical side, you could think about, how can you come up with a personalized therapy. And I'm sure this implication rules gives us a very simplistic view of the diversity that we see in the clinical side. And among the diversity, there is many different groups, and you can form these groups based on Boolean logic, and that gives you a personalized situation for a patient, and you can see which patient will benefit. And you saw the CDX2 story. CDX2 negative group benefits, so that's like really personalized, right? So CDX2 positive group do not benefit from chemotherapy, so you only give it to CDX2 negative stage 2 colon cancer.
Thank you. I found that like a really interesting point of the talk.
Are there any more questions?
You can always reach out to me by email and ask any question if you have. These are very simple techniques, and all of you can do it very easily. I'm sure there's already a lot of data out there to look at relationship like this. Which relationship is fundamental, right? Nobody asks this kind of questions. Which is invariant? And so these are really important questions that can give us interesting insights into diseases.
Thank you so much, Dr. Sahoo. So that would wrap up the speaker session.